Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. Our lecture today is about translation or protein synthesis, that it is complement of what our started in the previous two lectures on molecular genetics.
أهداف هذه المحاضرة هي كالتالي: Identify the genetic code and explain how it's read. Summarize the process of translation on free and attached ribosome to rough endoplasmic reticulum, and explain how most eukaryotic genes are regulated.
The genetic codes is the triplet sequence of messenger RNA that specify of the certain amino acids. Each codon is made up of 3 bases, such as CCU. There would be form 64 codons, more than enough to code for 20 amino acids. There are 61 codons code for the different types of the amino acids. AUG codons has many functions. It is not only code for the amino acid methionine, but also can provide a signal for the start of the polypeptide chain. Therefore, it is called start codon. There are 3 other codons. Do not codes for any amino acids, that it is called stop codon. The triplet codons characterized by: it is unambiguous. That means each codon specifies for the particular amino acids. For example, the codon ACG codes for the amino acid threonine and only threonine. It is non-overlapping. That means the triplet codons are adjacent. This means that the successive triplets are read in order. Each nucleotide is part of the only one triplet codon. 3. They are not separated by punctuation, such as comma. The genetic information transformed from the DNA into the messenger RNA by a process called transcription. Then the messenger RNA moves into the cytoplasm, and transfer of the information from the messenger RNA to the polypeptide, it is called translation. The proteins give the characteristic of an organism, or that called traits. Translation process is composed of 3 steps: initiation, elongation, and termination, as in replication and transcription. The translation begins when a small subunit ribosome binds to the messenger RNA near the AUG codon. AUG indicates the beginning of the gene, called start codon, that it is which is specified for the amino acids called methionine in the cytoplasm. This methionine is activated by the amino acids activated enzyme, then recognized and picked up by the tRNA to form methionine tRNA complex. At the end of the initiation, the large ribosomal subunit join to the small subunit. The second tRNA with amino acid bind to the second codon on the messenger RNA molecules at the A site. After that, the peptide bond formation. That means the amino acids in the A site forms bond with peptide in the B site. The tRNA with polypeptide in B site shift into the A site. Then the tRNA at the A site move to the B site and then exit. This process is repeated over and over again as the ribosome move along the messenger RNA chain and adding the amino acids to the growing polypeptide chain at the B site. The amino acids bind together by the peptide bonds and it is catalyzed by the enzyme called peptidyl transferase that it is found in the large ribosomal subunit. This phase of the translation called chain elongation. يمكن تلخيص الإلونجيشن كالتالي: بعد قراءة الريبوسوم للحامض الأميني أو الكودون المجاور للAUG تتحفز tRNA بتمييز والتقاط الحامض الأميني المناسب أو المطلوب، وبعد ذلك يرتبط إلى منطقة الA سايت. ثم تتكون الأواصر الببتيدية ما بين الحوامض الأمينية في منطقة الB سايت ثم تنتقل إلى الA سايت. بعد ذلك تحدث عملية الترانسلوكيشن، تنتقل tRNA مع البوليبيبتايد المتكون من الA سايت إلى الB سايت ومن ثم الخروج. هذه العملية تتكرر مرارا إلى أن يصل الريبوسوم إلى منطقة الستوب كودونز اللي موجودة بالترمينيشن of translation. The chain elongation is continuous until the ribosome encountered on the 3 possible stop codons. These codons do not code for any amino acids. Then the polypeptide is released and disassociated of the ribosomal subunits. Finally, certain protein must be altered before they can function. For example, in insulin hormone, as at the first translated as the polypeptide proinsulin, which made up of 80 amino acids. There are an enzyme cut it into the 51 amino acids and become functional. Some proteins must have sugars attached for them to become functional. This figure shows the summary of the protein synthesis. At the beginning, the genetic information transformed from the DNA into the RNA. The first type of the RNA, it is called messenger RNA, by a process called transcription. At the first form of primary messenger RNA, that it is contain sequence of non-coding sequence called introns and coding sequence called exons. This primary become mature by process called processing and modification splicing of the RNA. The mature RNA move through the pores into the cytoplasm, readily associated with small subunit of ribosome. At the AUG codon. AUG codon code for the amino acids called methionine. tRNA is activated to recognize and pick methionine from the cytoplasm and transfer into the messenger RNA. At the end of the initiation of elongation of the translation, large subunit attached to the small subunit. Then the ribosome move along the messenger RNA and read the adjacent codon, also activated of the tRNA to pick up the specific type of the amino acid from the cytoplasm and come into the A site on the ribosomes. This process repeats over and over until the ribosome reach to the stop codons. The translation is stopped and the ribosome disassociated and cleaved of the polypeptide chain. At the first, the polypeptide is non-functional, become functional when it is modified and transformed into other type of the functional polypeptide or proteins. Some of the proteins are synthesized on the free ribosomes in cytoplasm, and the others are made inside the endoplasmic reticulum. The synthesis of the proteins on the rough endoplasmic reticulum begins with messenger RNA leaves the nucleus and then move into the cytoplasm. Messenger RNA associated with ribosome or form of polysome. That means the individual ribosomes are held together by common strand of messenger RNA and then attached to the membranes of the endoplasmic reticulum by assist of proteins called ribophorines. The first sequence of the amino acid assembled called signal sequence. It is able to penetrate the membrane of the rough endoplasmic reticulum, and the ribosome continuous reading of the messenger RNA to form secretory protein. Then the secretory protein injected into the lumen of the rough endoplasmic reticulum, and the signal sequence enzymatically cleaved from the newly synthesized protein molecule. After that, budding from the rough endoplasmic reticulum as a vesicle and attached to the rough Golgi apparatus in which modified, and finally budding off as a vesicles and exit outside of the cell by exocytosis process. Question: what are the complementary sequences of the messenger RNA, tRNA, and protein of the following? This sequence of DNA. The complementary sequence of messenger RNA, this. And this, the sequence of tRNA and amino acids. The cells don't need the same enzymes and possibly other proteins all the time. For example, in the mammalian cells lining the gut produce a lot of digestive enzymes, while the muscle cells need a contractile proteins, actin and myosin. These enzymes or proteins controlled by genes, but they are expressed only in the certain cells of the body. Therefore, the eukaryotic cells must have a way to regulate the action of the genes. There are 6 primary levels for the control the activity of the genes. First, chromatin accessibility. In chromatin accessibility, determined which the gene is transcribed to form messenger RNA. Second, transcriptional control. In transcriptional control, determine which of a particular gene will be transcribed. Third, transcript processing control, in which determined which the messenger RNA is modified and become mature. Other control, transport control. In transport control, determined which the mature RNA is shipped out to the nucleus, from the nucleus into the cytoplasm. Translation control. In translation control, govern which of the messenger RNA molecule that reach to the cytoplasm and translated. Finally, post-translational control. In this, the control how the polypeptide become functional by modification. All of these levels of the control of the gene active determined which genes are expressed to form functional polypeptides. Cancer results from divided of the cells and control that it is can result from the mutation in the genes whose protein products regulate the cell cycle. There are two types of the normal cellular genes in human, either normal regulatory genes and DNA repair genes. The normal regulatory genes including the growth-promoting proto-oncogene and growth-inhibiting tumor suppressor gene and gene-regulate apoptosis. The mutation can change the proto-oncogene into the oncogene. Proto-oncogene, that means the normal gene that promotes cell division, which cause the cells to divide excessively. The other genes, called tumor suppressor gene, that it is normally act to inhibit cell division. If mutated, that cause cell to divide excessively also. Any mutated gene will be either repaired by DNA repair gene or killed by gene called apoptosis gene. If the genes not repaired and not killed by apoptosis, that it is cause cell divided excessively and cause cancer. شكرا لكم.